Sorry, I can't hear you. Sorry, I'm not audible.
Oh, hello. हाँ
ठीक है, अब को बने रहना, कॉल में बताता हूँ मैं, ठीक है।
हाँ हाँ हाँ।
हाँ मैम, अब देखना मैंने चेंज किए हैं ये वाले।
हाँ ठीक है सर, हम फिर बताएंगे।
ओके ओके। सो राइबोसोम मैंने बताया था 2 टाइप के होते हैं। एक तो आपका एंडो आर्या, और दूसरा हो जाएगा आपका एसईआर। ठीक है, तो आरईआर और एसईआर ये 2 टाइप के आपके पास एंडोप्लाज्मिक रेटिकुलम होते हैं। आरईआर का जहाँ तक है उसका फंक्शन होता है प्रोटीन सिंथेसिस करना। आरईआर का फंक्शन क्या हो जाएगा? प्रोटीन का सिंथेसिस करना। तो आरईआर जो है वो करेगा आपका प्रोटीन सिंथेसिस, और एसईआर जो है वो करेगा आपका लिपिड सिंथेसिस। ठीक है, क्योंकि इसके पास कोई भी एसईआर के पास कोई भी राइबोसोम नहीं होता है, तो इसलिए प्रोटीन सिंथेसिस नहीं करेगा। ठीक है, ओके। सो अगेन मैं बताया था कि साइटोप्लाज्म को आरईआर 2 पार्ट में डिवाइड कर देगा, और आरईआर बेसिकली एंडोप्लाज्मिक रेटिकुलम 2 पार्ट में डिवाइड करेगा। एक तो होगा आपका एक्स्ट्रा ल्यूमिनल और इंट्रा ल्यूमिनल। ठीक है, आपके पास क्या है? साइटोप्लाज्म है। साइटोप्लाज्म को जो 2 पार्ट होंगे, एक तो साइटोप्लाज्म वो जो कि आपका एंडोप्लाज्मिक रेटिकुलम के अंदर है, और दूसरा एंडोप्लाज्मिक रेटिकुलम के बाहर है। ठीक है, तो नाउ द साइट जो आपका साइटोप्लाज्म में 2 पार्ट में डिवाइडेड हो गया, एक तो ल्यूमिनल और एक्स्ट्रा ल्यूमिनल। ल्यूमिनल और एक्स्ट्रा ल्यूमिनल। ठीक है, ल्यूमिनल में जो होगा वो आपका ल्यूमिनल में क्या होगा? इनसाइड दी एंडोप्लाज्मिक रेटिकुलम। ठीक है, तो ये जो इनर साइड है, ठीक है, ये इनर साइड है वो है इंट्रा ल्यूमिनल। इंट्रा ल्यूमिनल। जो आउटर साइड है साइटोप्लाज्म एंडोप्लाज्मिक रेटिकुलम के, उसको हम बोलते हैं एक्स्ट्रा ल्यूमिनल। एक्स्ट्रा luminal. So intraluminal and extraluminal. Endoplasmic reticulum divides the cytoplasm into two species. One is the intraluminal and second is the extraluminal. ठीक है, intraluminal inside the ER, inside the ER, और दूसरा होगा आपका एक्सट्राल्यूमिनल, मतलब कि आउटसाइड द ER, आउटसाइड ER। ठीक है, सो दो अपने एंडोप्लाज्मिक रेटिकुलम कितने टाइप के हैं? दो टाइप के होते हैं। RER, RER हैव द राइबोसोम on its surface, and SER, they do not have ribosome, because the RER have the ribosome, and as we know that ribosome is a protein factory. Ribosome is a protein factory, मतलब कि राइबोसोम जो है वो क्या करता है? इट विल सिंथेसाइज द प्रोटीन। सो राइबोसोम विल सिंथेसाइज protein, will synthesize the protein. So basically ribosome synthesize the protein. So RER का फंक्शन क्या हो जाएगा? प्रोटीन सिंथेसिस। And SER will do not have the ribosome, so it will not synthesize the protein. Instead of the protein, it will synthesize the lipid-like steroidal hormone. Sorry, this one. It will synthesize the lipid-like steroidal hormone. Okay, lipid-like steroidal hormone. Okay. Now RER is present in the cells. It present in the cells which are actively involved in synthesis of protein, ठीक है। तो लार्ज अमाउंट में हमको RER कौन सी सेल्स में मिलेंगे? ऐसी सेल्स जिनके अंदर की आपको प्रोटीन सिंथेसिस करना है और सिक्रेशन करना है, तो वहां पर हमको क्या मिलेगा? RER मिलेगा, वहां पर हमको RER मिलेगा। ठीक है, तो SER के बारे में आपको बताया गया कि SER में क्या मिलता है आपका। SER में सिंथेसाइज होता है लिपिड लाइक स्टेरॉइडल हार्मोन। Now second organelle of the endomembrane system is the Golgi apparatus. Golgi apparatus. So this Golgi apparatus is just adjacent to the endoplasmic reticulum. ठीक है, मान लीजिए आपकी एक सेल है, ठीक है, इस सेल में आपका सेंट्रल न्यूक्लियस प्लेस होगा। मान लेते हैं कि ये एनिमल सेल है, ठीक है। एनिमल सेल में जो न्यूक्लियस होता है वो सेंट्रली प्लेस्ड होता है, तो ये न्यूक्लियस सेंट्रली प्लेस्ड हो गया, और यहाँ पर आपका ये एंडोप्लाज्मिक रेटिकुलम हो गया, और जस्ट एंडोप्लाज्मिक रेटिकुलम के नीचे आपका गोल्गी एपेटस होगा। ठीक है, ये क्या है? आपका गोल्गी एपेटस है। ठीक है, इसी तरह गोल्गी एपेटस और गोल्गी कॉम्प्लेक्स। सो गोल्गी कॉम्प्लेक्स first observed by the Camillo Golgi. Golgi in 1898. They contain the many flat-like. If I will draw the individual structure of the Golgi apparatus, then it will look like this. It will look like this. Okay. Okay. So they are the disc-like, so disc-shaped or sac-shaped cisterns. Okay, they are the many flat disc-shaped, disc-shaped, flat disc-shaped, or sac or cisterny. Each, each sac, this each sac is known as the cistern. Each sac is known as the cistern. This one is known as the cistern. One cistern is second, third, fourth, fifth. Okay, so this is the flat disc-shaped sac or cisterni. This is known as the cisterni. Please remember this name, cisterni. It is present in the Golgi apparatus, and one another thing, that is the cristae. Cristae, it is present in the mitochondria. Okay, cristae is present in the mitochondria and cisterni present in the Golgi apparatus. Diameter of the diameter of the sac or cisterni is 0.5 micrometer to 1.0 micrometer. Okay, and these stacks, they are arranged parallel to each other. They are arranged parallel to each other. So cisterns arranged parallel to each other. Okay, so there's varied number of cisternae may be present in different type of the cells. Okay, so the number of the cisternae is not specific. Okay, किसी भी सेल का जो सिस्टर्नी में नंबर होता है, तो सिस्टर्नी का नंबर होता है किसी भी गोल्गी बॉडी में, वो स्पेसिफिक नहीं है. It will vary cell to cell. Okay, now system जो Golgi apparatus has two phase. Golgi apparatus has two phase. Please pay attention here. This phase which is towards the nucleus. This phase which is towards the nucleus. This is the nucleus. Okay, this is the nucleus. Yeah, this is the nucleus, and this phase is towards the nucleus. Okay, assume this. Assume I am drawing the nucleus here. This is the nucleus, and the phase which is towards the nucleus is known as the cis phase. Is it known as the cis phase or forming phase? The phase towards the periphery. Towards the periphery means towards the cell membrane. This phase is known as the trans phase or maturing phase. Maturing phase. ठीक है. So the Golgi complex or Golgi body has two phases, cis phase and forming phase. Cis phase or forming phase, or trans phase or maturing phase. Now the name of the phase is depends on the function of the part of the Golgi body. What is the function of the cis phase or forming phase? So just see here. Here I'm drawing a cell, and we have nucleus here, and adjacent to the nucleus we have the endoplasmic reticulum. Okay. Now, endoplasmic reticulum will synthesize its product in the form of the vesicle. It will synthesize in the form of vesicle. So we have some vesicle. Now this vesicle will, so we have endoplasmic reticulum. It will form its product in the form of the vesicle. This vesicle will fuse with the vesicle fuse with the Golgi body, the Golgi body, and undergo packaging. Okay, so it will form with the, this will form with, this will fuse with the, sorry, this will fuse with the Golgi body. Okay, so these are the, this structure you can see here. They are just fusion of vesicle with the Golgi apparatus. So this vesicle fuse with the Golgi body and it will synthesize, sorry, it will package the synthesis of endoplasmic reticulum, packaging in the form of the lysosome. So that's why this is the forming phase. It will be it will form the product, or it will correct the any uh, इंटरप्शन एनी डिफेक्ट इन दी प्रोडक्ट. जो भी प्रोडक्ट इसने बनाया है उसमें कोई डिफेक्ट होगा तो वो करेक्ट करेगा, उसको पैकेज करेगा. पैकेज करने के बाद में जब इस पॉइंट तक आएगा, इस फेज तक आएगा, तो इट विल मैच्योर इनफ नाउ, ये मैच्योर हो जाएगा और इट विल ट्रांसफर दी प्रोडक्ट टू दी साइटोप्लाज्म और अदर टारगेट सेल. तो ये जो मैच्योरेशन है और ट्रांसफेज है, ये किसके फॉर्म में आपका किसके फॉर्म में रिलीज करेगा इसके प्रोडक्ट को? इट विल रिलीज इन द फॉर्म ऑफ द लाइसोसोम. लाइसोसोम, ठीक है. ओके, सो हाँ, तो किसके फॉर्म में सिंथेसाइजेशन हो चुका है? लाइसोसोम के फॉर्म में सिंथेसिस हो चुका है. अब ये जो सिंथेसिस है वो आपका दोनों का फंक्शन समझ में आ गया होगा आपको, ठीक है. दोनों का फंक्शन क्या है? देखो, गोल्गी एपेटस का और एंडोप्लाज्मिक रेटिकुलम का फंक्शन क्या था? एंडोप्लाज्मिक रेटिकुलम को क्या था, मैंने बताया था आपको. एंडोप्लाज्मिक रेटिकुलम इज द many tubular like structure in the cytoplasm. They are the tubular like structure in the cytoplasm. This tubulars, this tubules basically, this tubules will this tubule will divide the cytoplasmic space into two compartments. One is the inner compartment, which is this is the inner compartment, okay, and one is the outer compartment, the cytoplasm. This is the outer compartment in the cytoplasm. So the inner compartment is known as the intraluminal, or the external compartment, or external compartment is known as the extraluminal. ठीक है, so intraluminal, extraluminal. Now Golgi apparatus, I told you about the Golgi apparatus, his phase, maturing phase, and the synthesis packaging. Now function of the Golgi apparatus, I told you the function of the Golgi apparatus is the function of Golgi apparatus Okay, so function, first function is the packaging. Okay, or formation of lysosome, formation of lysosomes. ठीक है, formation of lysosomes. It will also help in the formation of glycoprotein and glycolipids. Formation of glycoprotein and glycolipid. Please remember this thing for NEET exam. Formation of glycolipids and glycoproteins. Okay, so it is about the Golgi apparatus. Now, third thing in the mm, endomembrane system is the lysosome. So, what is the lysosome? Lysosome, it is the packaged form. Okay, lysosome, it is the packaged form or package form membrane bound vesicular structure formed formed due to the packaging in Golgi apparatus और गोल्गी बॉडी. तो गोल्गी बॉडी में पैकेजिंग होने से जो सिंथेसिस हुआ है उसको हम बोलते हैं लाइसोसोम. नाउ also known as also called as the suicidal bag of the cell. Suicidal bag of the cell. Why so? भी देखते हैं क्यों बोलते हैं इसको. Okay, so assume that we have this lysosome. Lysosome is a vesicular structure. Vesicles means a fluid filled cavity. Vesicles means a fluid filled cavity. So this is a vesicle, and this vesicle we have. This vesicle is filled with the many enzymes. This vesicle is filled with the many enzymes, and these enzymes are known as the hydrolytic enzyme. The enzymes are known as the hydrolytic enzyme. Different type of the hydrolytic enzymes are present there. Okay, so lysosome. Sorry. So we have enzymes known as the digestive enzyme, also known as the hydrolytic enzyme. The word lytic means breakdown. So these enzyme are different type of the enzymes are there, like nucleases, lipases, carboxylases, carbohydrases, okay. So they are the enzymes. These enzymes will act on the substrate. What is the substrate for the nucleases? The substrate for the nucleases enzyme is nucleic acid. Nucleic acid is the substrate for the enzyme nucleases. Lipase is the lipid is the substrate for the lipase enzyme. Carbohydrates is the carbohydrate. Carbohydrates is the substrate for the carbohydrates. Okay, so when they will act on the nucleic acid, so they will break down the nucleic acid. When they will act, or one more enzyme is there, protease. Protease also they act on the protein. They will act on the protein. All the enzyme will act on its substrate and they will break down the substrate. They will break down the substrate. Now, please see here and pay some attention. We learned about the membrane. We learned about the plasma membrane. In the plasma membrane is made up of the lipid and protein. So by anyhow, okay. So this is the plasma membrane. This is the plasma membrane, and this plasma membrane is made up of the lipid and protein. Okay, and in the plasma membrane we have the lysosome. So anyhow, assume that lysosome are broken down. They are broken down. So the content of the lysosome will come out. The content of the lysosome will come out, and you know that the content of the lysosome is a hydrolytic enzyme. The content of the lysosome is the hydrolytic enzyme. So hydrolytic enzyme like protease, it will act on the protein. Okay. Second thing, lipase will act on the lipid, and when the protein will break down, lipid will broken down, so the cell membrane also broken down, and it will look like a suicide. Okay, cell suicide. It will look like a cell suicide. I think it is clear to you why the lysosome is known as the suicidal bag of the cell. Please remember about the enzymes. Most important enzymes. Sorry, most important are the enzymes are important. These enzymes are important, and the other name for the lysosome and the site. Basically, Golgi apparatus helps in the formation of the lysosome. Okay. Now, fourth part of the endomembrane system is the vacuole. So, vacuole. As you know, vacuole is the membrane-bound space found in the cytoplasm. It is the membrane-bound space found in cytoplasm. Membrane-bound space found in cytoplasm. Now, plant cell has the vacuole. Plant cell has the vacuole. Animal cell may or may not have. Animal cell may be or may not. But if present, but if it is present in animal cell, then the size is very small. Size is very small in the animal cell. If you talk about the vacuole in the plant cell, then 90% of the space or volume of the cell, but in plant cell, 90% volume of cell is is occupied by the vacuole. Simple. ठीक है 90% of the volume is occupied by the vacuole. Please see here, that's why the okay, so this is the your plant cell is general plant cell is rectangular in shape. Okay, and this one is your vacuole, and here we have the nucleus. So nucleus is acentric. Why it is acentric? Because of the vacuole, because vacuole is bigger in size. That's why it will push the nucleus towards the corner. So it is acentric. While in the animal cell, I told you, and in an animal cell, almost all animal cell have the centrally placed nucleus. Okay. So 90% of the space is occupied by the uh, vacuole in the plant cell, and the membrane of the vacuole is known as the tonoplast. Please remember this one. Membrane of the vacuole is known as the tonoplast. Okay. Okay. Now, so the tonoplasm membrane, it facilitates the transportation of many material against the concentration gradient. Against the concentration gradient. What is the concentration gradient? Concentration gradient means difference. Okay. और द कंसंट्रेशन ग्रेडियंट एक्चुअली मान लेते हैं हमारे पास दो ग्लास है पानी से भरे हुए, ठीक है। ग्लास ये है, वी हैव टू ग्लास filled with the water. One glass is this and second one is here. Okay, one is filled with the both the glass is filled with the water. Both the glass filled with the water, and but up to different limits. Assume that if I connected them with a tube, then both will achieve a equal limit. Both will achieve a equal limit. So water will transport from A to only this one is A, this one is B. It will transport from A to B. But if I want to transport the water from this region to this region, okay, this is the against the gradient. Okay, according to the difference, the water should be transported from the higher level to the lower concentration. Higher concentration to the lower concentration. Water should be transported from the higher concentration to the lower concentration. But if you want to reverse this direction from lower to higher concentration, that means it is the against the gradient. This is the against the gradient. So when you want to transport the water or transport any material against the gradient, you need some energy in the form of the ATP. So transportation of many material against the concentration gradient in the vacuole. So in the vacuole there is the many minerals or many material which is more than in the cytoplasm, because they are transporting against the gradient. Okay. Now we have types of vacuole, like first is the contractile vacuole, and second one is the food vacuole. So food vacuole is present in the protista due to engulf of the food, and contractile vacuole it is present in amoeba. For the osmoregulation and the excretion. So please remember about the contractile vacuole and the food vacuole. They are different from the plant vacuole present in the cells. Okay. Now next, cell organelle. So we completed the endomembrane system. Endomembrane system consists of the 4 cell organelle. One is the endoplasmic reticulum. Second is the Golgi apparatus. Third one is the lysosome, and fourth one is the vacuole. Okay. Now mitochondria. Okay, so mitochondria first question for the basically mitochondria. You can see under the mitochondria after staining. Staining is important. Staining is the coloring. Okay, when you prepare the slide uh, for the microscope observation, you have to stain the slide. You have to colorize the slide by different type of the staining dyes. There is methylene blue, saffron dyes are there. I think you know about it in your uh, biology practicals are there on the basis of slide preparation and all that. Okay, so staining is important for the important to see the mitochondria under the microscope. Now, first question which is for the NEET, number of the mitochondria is depends on the physiological activity of the cell. So, number of mitochondria is depends on the physiological activity of the cell. The number of mitochondria depend on the physiological activity of the cell. Okay, please remember this statement. Number, size, and shape. All the properties variable. All the properties variable, and the typically the mitochondria is a sausage shaped or cylindrical. It is sausage shape. Shape is sausage shape or cylindrical shape. Okay, having with the diameter 0.22110 micrometer the diameter, and length is 0.1.0 to 4.4.1 micrometer is the length. Okay, so length and diameter is variable according to the cell. Okay, next. Okay, so please assume that this is your mitochondria, and mitochondria has the sausage shape. Okay, mitochondria. First thing about mitochondria, the most important thing you can't afford to forget it. You can't afford to forget it. First thing is it is डबल मेम्ब्रेनस स्ट्रक्चर. ठीक है, आप कभी मत भूलना प्लीज इसको, डबल मेम्ब्रेनस स्ट्रक्चर है ये. सेकंड थिंग, इट हैज sorry for interruption. हाँ, second thing about the second thing, first thing is you can't forget about it. That double member structure. Its own DNA. It's on DNA. That's why it is known as the semi-autonomous organism. It can divide on itself. Semi-autonomous. Please remember this thing. Most important thing in the mitochondria. Okay, now I told you it is double membrane structure. So it is the outer mitochondrial membrane. Outer mitochondrial membrane. And you have another, that the inner mitochondrial membrane. Okay. This inner mitochondrial membrane shows infolding. Okay, shows the infolding. This is the inner mitochondrial membrane. This is the inner mitochondrial membrane. It shows the infolding. You can see these infoldings are there. Okay, these are the infolding. Okay, infolding of the inner mitochondrial membrane. And these inner mitochondrial membrane infoldings are known as the cristae folding. So this infolding is known as the cristae. Remember about it, cristae and cisternae. Don't confuse between them. Cristae is present in the mitochondria and cisternae is present in the Golgi apparatus. Okay. Now we have the outer mitochondrial membrane, inner mitochondrial membrane. Cristae is the infolding, and inside the cristae we have the matrix. Inside the cristae we have the matrix. Matrix is the fluid inside the fluid substance inside the infoldings. So the infoldings have the fluid, and this fluid is known as the matrix. And this is the intermembrane space. Intermembrane space. Okay. Now, what is the function of function? It is the site for. It is the site for cellular respiration. Okay, as you know, the statement about the mitochondria, that mitochondria is the powerhouse of the cell. So why the mitochondria is powerhouse of the cell? Because why the mitochondria is the powerhouse of the cell? Because mitochondria is the site for the cellular respiration. In the cellular respiration, there is the formation of ATP. Formation of ATP or ATP synthesis is there. And ATP is the energy currency of body. Energy currency of body. What the ATP stands for? It stands for the adenosine triphosphate. ATP stands for the adenosine triphosphate, and it is the energy currency of the body. That's why the mitochondria is the powerhouse of the cell. And mitochondria contain DNA. It is the single circular DNA. Single. Please remember this, single circular DNA is present in the mitochondria. Okay. So let's see once again about the mitochondria. There are the different thing about the mitochondria. Structure, number of the mitochondria depends on the physiological activity. Number, size and shape is variable. Shape is sausage shape or cylindrical shape. The diameter and length The structure of the mitochondria, it has the two membrane, outer membrane, inner membrane. Intermembrane space, then inner membrane shows infolding, and these infoldings are known as the cristae, and this cristae is filled with the fluid material, and this fluid material known as the matrix. Function is the site for the formation of ATP or cellular respiration. Okay, now next thing is the plastid. Okay, next thing is the plastid. Plastid is present. Plastid basically plastid is pigment containing cell organ. Pigment containing cell organelles. Pigment means color. Okay, the cell organelle which provide the color for the plants is known as the plastid. Now, human being or animal cell do not have any plastid in their cell because human being or any animal, they do not have different color. Obviously, they have the different range of the melanin. Okay, the range of the, so there is the melanin which is responsible for the skin color in the human being. Melanin is responsible for the skin color in human being, but in plant there is the plastid which is responsible for the different color of the leaves and different color of the plants. So pigment is the color containing or pigment containing cell organelle inside the plant cell. Also in the ignanoids, plant cell plus euglenoids. You will see about the ignanoids in the plant classification or biological classification. But for now, please remember the example in which the plastid is present. Now, plastid is of different type on the basis of the pigment. Plastid types depends on pigment. Depends on the types of pigment. Pigment they contain. Like if they contain green color pigment known as the chloroplast. If they contain different color except the green, colors except the green known as the chromoplast. Chromoplast. If they are the colorless, then they are known as the leucoplast. So leucoplast has the function of the storage because they are not providing any color. That means they are not waste material for the cell. No, they provide the function of the storage. Like we have three types of the leucoplast. One is the aleuroplast, elioplast, and amyloplast. So aleuroplast, it will store the protein. Elioplast, oil and lipid storage. And amylo word means carbohydrate. So it will carbohydrate or starch. So it will store the starch. Okay. So we have three type of the plastid. One is the chloroplast, green color containing pigment. Chromoplast, a color except the green color and colorless. Leuco means white. Okay, leuco means you know that white, but white in the botany is colorless. Okay, white is not color here; it is colorless. Okay, now we will see about the chloroplast. Chloroplast, the site for the photosynthesis. Chloroplast is the site for photosynthesis. Chloroplast. Chloroplast again. two important thing, chloroplast or plastid basically. Green color containing plastid. It contains the chlorophyll plus carotenoids. Carotenoids is orange red color pigment, and chlorophyll is the green color pigment. It is the orange yellow color pigment. Okay, so chloroplast containing two pigments, one is the chlorophyll and carotenoids. Okay, now the structure of the plastid is usually similar for chloroplast, chromoplast, or the leucoplast. So in the chloroplast, it is the double membranous structure. It is the double membranous structure. The double membranous structure, it will occupy or enclose a space. The double membrane encloses a space, and the space is known as the stroma. So this double membrane, the chloroplast encloses a space, and this space is known as the stroma, and the stroma contains the different structure. Like, so this is the outer membrane, obviously inner membrane, no doubt, and this membrane, this stroma contains structure known as the thylakoids. They are the thylakoids. They are the thyla. Okay, single structure in thylakoid. This is the, now please concentrate here. This one, the single structure. This is the thylakoid. But the coin of piles of coins. This is the piles of coin like structure. Piles of coins like structure complete. Is known as the granum. This is the granum. Granum. Granum is a singular word, or grana is the plural. Okay, so complete under the grana. These grana are connected by a linkage, and this linkage are known as the stroma lamellae. Stroma lamellae, and the single individual of the granum is known as the thylakoid. Thylakoid. Okay, so this is the structure of chloroplast. Here the arrangement of the thylakoid like piles of coin known as the granum, and thylakoid have the, okay, this is the site for the site, chloroplast the site for the photosynthesis. Please remember this. It will, we will see about the chloroplast again in the plant physiology. Okay, when we will start the plant physiology, we will again see about the site chloroplast, because this is site of the photosynthesis. How the process of photosynthesis will takes place here, we will see it in the plant physiology. Okay, एक चीज तो ये हो गई. So chloroplast is complete, plastid is complete, and okay, one more thing. Here the DNA, here the DNA is present. Okay, same thing is in the mitochondria, but DNA is different. DNA, it is double stranded circular DNA. Okay, double stranded circular DNA is present, and that's what is known the semi-autonomous. Semi-autonomous means, semi means half, autonomous means self-directed. Okay, so they will divide on their self. That's what means the semi-autonomous. So DNA is present, double membranous structure is there. Okay. Next is the ribosome. Next is the ribosome. So what is the ribosome? Ribosomes are of two types. Ribosomes are of two types. They are the granular structure. There are two types. One is the bound ribosome. Another one is the free ribosome. The wild ribosome or bound ribosome are the one ribosome which is bound to the ER or other endoplazmic reticulum or other cell organelle. Free ribosomes they are freely living in the cytoplasm. Okay. Now you know that the ribosome consists of two subunits. Ribosome it consists of two subunits like a small subunit or large subunit. Okay. Now again we have two type of these ribosome. Prokaryotic like 70S, which is found by 50S and 30S. 50S is the larger subunit and 30S is the smaller subunit. Why stand S stands for the Sedberg unit. Sedberg unit. It measures the density and size of the ribosome. Measure of density and size of ribosome. Also known as the sedimentation unit. Sedimentation unit. Sedimentation unit. Okay, so prokaryotic ribosome is a 70S ribosome. It consists of subunits 50S, 30S. 50S is the larger, 30S is the smaller. Eukaryotic. Eukaryotic is the 80S ribosome. Consists of two 40S, 60S. 40S again larger, 40S is the smaller one. Okay. Function of the ribosome: synthesis of protein. Function is synthesis of protein. Simple. Okay. Synthesis of protein. Clear? Okay. So next topic we are going to discuss is the nucleus. Okay. So nucleus, it is first described by the Robert Brown. First described by the Robert Brown. First described by Robert Brown. Okay. Later the nucleus material of nucleus is stand by Fleming. And this material is known as the chromatin, given the name chromatin. Okay. Now here we are saying about the interphase nucleus. What is the interphase nucleus? When the nucleus of the cell, it is in which phase it is not dividing. Interphase nucleus is the nucleus of cell nucleus of cell in which it is not dividing. Simple. ठीक है, जब सेल डिवाइड नहीं करती है उसके न्यूक्लियस को हम बोलते हैं इंटरफेस। बेसिकली दो सेल होते हैं किसी भी सेल डिवीजन के, एक होता है इंटरफेस दैट इज द in which cell will prepare for the division, and one is the M phase, the phase in which the cell will actually divide. So we will start the interphasic nucleus. Okay, so basically see here what is the nucleus. Here you will have, okay, please concentrate here how I am drawing the nucleus. Collects, you can try to collect some information from the structure. So nucleus is a double membranous structure. First of all, nucleus is a double membranous structure. Okay, there is the matrix, which is the nucleoprotein. Inside the nucleus, nucleoprotein. Inside the nucleus, a spherical body. Spherical body inside the nucleus. Okay, and okay, so much things are there. Okay, madam. Okay, and now please concentrate here. Okay, sorry. There is the fusion of these two membrane. Fusion of these two membrane. Fusion of these two membrane leading to form nuclear pore. They will provide us nuclear pore. Okay. So here we have the, you can see here now clearly. Okay, you can see the clear structure of the nucleus. We have the matrix. Okay, we have the matrix inside the nuclear membrane, and this matrix is known as the nucleoplasm. Known as the nucleoplasm. Okay, simple thing. This is the chromatin material. This is the chromatin material. It contains nucleoprotein. Chromatin material consists of the nucleoprotein. This is the nuclear membrane, outer and inner. And due to the fusion of the nuclear membrane, we get nuclear pore. Nuclear pore. This is the spherical body spherical body inside the nucleus known as the nucleolus. Okay, so a spherical body inside the nucleus known as the nucleolus. Chromatin material consists of the nucleoprotein, nucleoplasm, nuclear pore. How the nuclear pore is formed? Due to the fusion of nuclear membrane. Nuclear membrane. Okay. What is the function of the nuclear pore? Okay. One most important thing is there. The space between these two nuclear membrane is known as the perinuclear space. Perinuclear space. Okay. An outer membrane of the this outer membrane again see here this outer membrane continues with the endoplasmic reticulum. I told you this will continue with the endoplasmic sorry it will continue with the endoplasmic reticulum. It will continue with the endoplasmic reticulum. So outer membrane will continue with the endoplasmic reticulum. Nuclear pore will provide the function for the passage of RNA protein molecule in body direction. So nuclear pore it will provide passage for movement of the RNA and protein in both direction. In both direction. Okay, so this is about the again see here nucleus. Okay, okay. One more thing about the nucleolus. The nucleolus is the organelle which is without the membrane. It is without the membrane, like ribosome. Ribosome also organelle which is without the membrane. Where the ribosome I told you. Okay, there is no membrane are there. Please remember it. No membrane in the ribosome plus no membrane in the nucleolus. They are continuous with the nucleoplasm. No membrane. I told you about it. Yeah. Without the membrane. Without the. Please keep this point in your mind. Function of the nucleosome is the ribosomal RNA. Synthesis of ribosomal RNA. Okay, ribosomal RNA. So please just you can just draw the diagram and you can write it all the things. So it is easy way to remember the things. आप आराम से इसको याद कर सकते हो इजी वे में आप इसको मेमोराइज कर सकते हो क्योंकि सब कुछ आपके एक ही डायग्राम में आ जाएगा ठीक है क्योंकि एग्जाम में तो लिखना नहीं है आपको आप क्या करना है नीट के प्रिपरेशन करना है now we will see about the chromosomes okay so I think it is clear up to the what nucleus okay nucleus is clear and the ribosome is clear mitochondria and the endomembranous system. Now we will study about the chromosomes. Okay, what are the chromosomes? How they are formed? Okay. So chromosomes are formed due to the condensation of the chromatin material. So chromosomes, they are formed due to the condensation of chromatin material. Now assume that we have a basically DNA is the condensed form of the DNA. So we have the DNA. From the DNA, it will package in the form of the nucleosome. So it will package in the form of the nucleosome. Then condensation will be there like this. Okay. DNA will wrap around the histone protein. I told you about it. So packaging of the DNA is there. Then it will turn into form the chromatin material. Then chromatin material again it will condense, more condensation form of the chromatin material, and then finally we will get what? Chromosome. Okay, so okay, so we will get a perfect chromosome. Basically, chromosome it is a condensed form of the chromatin material. Now, some okay, this chromosome contains the histone protein plus DNA, some non-histone protein also, and also RNA. Okay. Now, every chromosome essentially has a primary construction. Okay. So, first of all, the chromosome does not look like this. Chromosome starting में ऐसा नहीं दिखता है. Chromosome is simply like this. It is simply like this. But after some time, there is the primary constriction. Primary constriction. So, it will constrict. Okay. Let me draw. So primary, the chromosome is like this. Then there is the primary constriction. What is the primary constriction? Strict from the both the side, from both the side. Like it will constrict from here, and it will also constrict from here. And this is known as the primary constriction. Now chromosome will look like this. So it is the primary constriction. Okay, primary constriction. So this primary constriction is known as the centrosome. This is the the centromere, no, centromere, and primary constriction is known as the centromere. Okay, so this is the centromere. Okay, centromere, and this centromere is the primary constriction. Okay, primary constriction. Okay, so from the chromosome, we will see the topic in the next lecture. I mean to say the next Sunday. So for now, please uh, revise all the things. Okay, and all the questions from the NCERT and other books. Thank you and have a nice day.